Hello, students. Welcome back to Bio Decode, where biology is decoded into steps. Today, we will dive into our cutting edge topic, multi-omics, the way scientists combine many layers of molecular data to understand how cell truly works and how the insight is transforming medicine. By the end of this video, you will understand how studying DNA, RNA, proteins, and metabolites together reveals the complete story of what's happening inside a cell. So, without any further delay, let's get started. Every discovery starts with a biological sample, single cell, tissue, or organism. Inside it, there is an entire world of molecules, includes DNA, RNA, proteins, and metabolites. So, scientists don't just look at one layer anymore. They have to explore all of them together to see the big picture. Where does this molecule journey begins? With the blueprints, the genome. Let's unlock the first layer of life, the DNA blueprint. The genome holds the complete DNA sequence, a master plan containing every gene. By sequencing DNA, scientists identify what genes are present, their mutation, and potential function. But knowing the plan is not enough. We need to know which genes are actually active. That's where our next layer comes in, the transcriptome. Let's move from what's possible in DNA, what's happening right now in RNA. Transcriptomes show all RNA molecules transcribed from DNA. It's like checking which page of the genetic book are currently being read. This helps us understand how cells respond to stress, infection, or environmental changes. But RNA only carries messages. We still need to see who is doing the actual work. So let's follow those instructions to the real worker inside the cell, the proteins. Proteomes includes all proteins within the cell, each with a specific function, interactions, and modifications. These proteins carry out cellular processes like metabolism, defense, and signaling. Studying the proteomes helps scientists understand how cell functions in health and fail in disease. But protein does not work alone. They rely on small molecules for energy and regulation. So let's move deeper into the cell's chemistry to the world of metabolites. Metabolomes captures all the small molecules like sugar, lipids, amino acids. They reflect the final outcomes of the biological reactions. The cell's real-time activity and shift in these molecules can indicate stress, disease, or adaptations. Now imagine combining all these molecule snapshots, what we get in true biological insight. Let's bring it all together into one integrated view. When scientists combine genomics, transcriptomics, proteomics, and metabolomics, and they create a full picture of how life operates, from codes to function to outcomes, like watching the script, the cast, the performance, and the review all at once. This is what makes multi-omics so powerful. But what we can actually do from this knowledge? Let's see its real world implications. Multi-omics has revolutionized medicines. It helps to detect disease earlier and understand treatment resistance and design the personalized therapies. In cancer research, for example, multi-omics data helps to identify which treatment works best for specific genetic profiles. It transforming diagnosis, treatment, and even preventive healthcare. So next time when you hear the word omics, remember It's the science of decoding life layers by layers. Multi-omics is the future of biological discovery, turning complex molecular data into actionable insight. Whether you are studying cell biology, toxicology, or human health, mastering this layered approach will put you ahead. If you want to note down the whole lecture, you can refer to these slides. I have summarized all the points I have spoken in whole lecture. And yes, students, don't forget to like, share, and subscribe if you like this video. See you in the next decoded lecture.